جهد غير عادي وجهد مشكور لكل السادة الزملاء المشاركين في هذه الورشة من الدكتور أشرف بكري والدكتور عبد الله بن ناصر وكل الزملاء في هذا العمل الذي يحسب لقسم الوراثة داخل كلية الزراعة جامعة عين شمس. في البداية بنرحب بأستاذنا الدكتور صلاح جلال أستاذ الإنتاج الحيواني لتشريفه لنا في هذا اليوم، وأرحب أيضا بأخي وحبيبي وزميلي الأستاذ الدكتور أشرف بكري والدكتور عبد الله عبد الناصر. أنا أقدم هذه المحاضرة ثم أستأذنكم لارتباطي بلجنة ممتحنين هي الآن منعقدة وثم بعد ذلك مجلس كلية. فسريعا كده. Dear Alex, dear colleague, dear students, good morning. It is nice to meet today to listen to Dr. Alex in bioinformatics. What is bioinformatics? Bioinformatics generally applies computer science to solve a biological problem. However, the field is interdisciplinary and constantly developing, so it is difficult to answer this question in one sentence. Bioinformatics became necessary as a part of our education. Dear Alexander Bonto, researcher at the Center of Research in Agriculture Genomics, Barcelona, Spain, will transfer his teaching experience in bioinformatics education in local and international university in his talk today. I wish you a good time and enjoy the lecture. Thank you and goodbye. Assalamu alaikum wa rahmatullahi wa barakatuh.
Barcelona, so Barcelona is not football only. Dr. Ponte, you know, he looks very young and he is young. Has been to many renowned places. He has been to King's College of London for genetics and medical genetics. He has been to University of Liège and INRA in Belgium. His research interests are very wide. I hope we could have the time to discuss some of it in our workshop. He's interested in animal genetics, human medical and domestic animal models for human diseases, and epigenetics, which is very interesting. Now he is accomplished scientist and research worker. He published some 27 papers in scientific journals and participated in many projects. And as mathematics unified physical sciences, we see now genetics is unifying biological science. And by the way, the BSc degree of Dr. Ponte is veterinary science, so he's a veterinarian by first degree, and his talk will be of animals and men. So women are out?
Yes.
Or just men?
Everyone, we are all the same.
Okay.
We are all the same.
All right. Now I should thank Dr. Abdullah Sharaf of the genetics department, who has been the force for processing the invitation of Alexander. Alexander, please go ahead.
Thank you very much. Is everyone hearing me here, or should I? Hi, everyone. Well, I will try to speak a bit loud. Maybe just one second. Hello? No. No problem. So first of all, I want to thank everyone for inviting me to give this talk today, and I in particular Abdullah for pushing me and trusting me that much what I see outside, I think it's much more than what I am.
Thank you
very much. Much more than what I really am. Well, Abdullah invited me to come and speak here about my research interests and the research I do. So I'm not going to speak about bioinformatics. I'm not going to speak about bioinformatics as a nature of teaching. I had my session 2 days ago. I'm just going to speak about the research, basically what I do. And that will be split in 2 parts. First, the part of what I have done until now, which is basically what explains what I want to do and why I want to do, and what I'm doing and programming for the future. So yes, as it was said, I will speak about animals and men, use of genomic tools to, I think I will stop this. Use of genomic tools for the dissection of complex traits. I stop this. Right. Use of genomic tools for the dissection of complex traits. I could have given any other name, but I think somehow this exemplifies what I want to do. Yes, I am a vet, so I did my PhD in Barcelona in pig genetics, because in Europe pig is the most important livestock animal, the most important meat consumption. So it was genetics, genomics on meat production traits in pigs. Then I moved to Belgium, where I did a postdoc in sheep genomics, basically very similar. And then I went to Scotland. I did 2 more years in technology transfer. Then I went to London and I worked in human genetics, in psoriasis, as you will see. And about 1.5 years ago, I joined CRAC, the Center for Research in Agricultural Genomics in Barcelona, to study animal genomics, but trying to open a line in how we can use livestock animals as models, spontaneous models. That means diseases that occur spontaneously in animals. You don't need to do transgenesis or anything to transfer that into. So those diseases that occur spontaneously in animals, how to study them and benefit human diseases. That could be an example. For example, if you have coronary disease in humans, and you see a similar disease in sheep or in cows or in pigs, how to study this, how to get this information from those animals, which allow you much more plasticity in the experimental design to manipulate, to make populations, to make pedigrees, to give a treatment, study this, and try to transfer these results for the human counterpart disease. So I have split this into parts. The first part is my previous research, what I have done, and I have put basically 2 examples that I think grossly summarize what I have done and as well justify what I want to do. And then I will speak about this present research and future research. First of all, I hope you can read it well. Can everyone read here? Yeah. So I went to Belgium and I did, grossly speaking, the identification of the genetic variant that causes hypermuscularity in the texel sheep. And that was published in Nature Genetics because it was a breakthrough in finding genetic variation with regulatory effects that affect phenotypes. And that mechanism was not described before. So very briefly, this animal here is a sheep, is not a pig. This animal is with these big buttocks. This sheep. It's called the Belgian texel sheep. It's an animal that has been highly selected with a strong selective pressure in the last 100 years for muscularity. So basically, milk production, wool, all this, it's really very bad. It's basically muscularity, and it's really a massive, it's the terminator, the Arnold Schwarzenegger of sheep. And what we did is what was done back on the mid-90s, late 90s, which was a QTL scan. Basically, you generate a pedigree in animals, an F2 pedigree in this case, meaning that you take animals from one population, that is a population that you really want to study, in this case the texel, the hypermuscle sheep, and animals from the other population, which have to be a population very different, in this case not muscular. We took another breed, we generated an F1, generated an F2. This resource that was about 300 sheep was then genotyped by about 150 microsatellites, typical experiment, and you do linkage mapping, QTL mapping. What we get here by doing this is identifying a region of the genome that affects the phenotype, that affects muscular hypertrophy. This region then was refined by selecting further animals that were recombinant in this region of the genome and adding more markers so that you have a region that is, let's say, 20 megabase pair. By doing this, we can refine it to 1 megabase pair. That was technology back from the 90s. Nowadays, few people use these technologies. Then when we had this region selected, the next step was to identify what is the causal variant. We found several genes, and there was a gene that was a very strong candidate, which is called myostatin, GDF8, growth and differentiation factor 8. And this gene is causing a very similar phenotype, and this has been proven in cows. So it was a very obvious candidate gene. What we did is to sequence that gene, and we didn't find any mutation in the coding sequence, in exon 1, 2, or 3 in the coding sequence, nothing, no aminoacidic change. What we found is well, maybe I should say that we genotyped this in hundreds of animals from more than 10 breeds. It was like 50 to 60 Texel sheep and maybe 60 to 70 animals from other breeds. The idea was that the polymorphism that causes the muscular hypertrophy should be one allele present in all the animals from that breed and absent in all the other animals from other breeds that are not hypermuscled. The concept is clear. So if something causes something, that allele must be present in all the animals that have it and absent in all that do not have it. So from all the variants we found, only one variant was accomplishing this characteristic, was a polymorphism in the 3' UTR. That is a region of the genome, of the gene, that is transcribed from genomic DNA to RNA, but not translated to protein. So you found it in the messenger RNA, but not in the protein. When we look at this region, we found out that this could be, for its sequence, would be a target site for microRNAs. Maybe. MicroRNAs are small RNAs, very small RNAs, 22 nucleotide long, that they are transcribed into microRNA, but not translated into protein, and their function is regulatory. They regulate the translation into protein of messenger RNAs. So you have the microRNAs, and they are navigating in the cell, and when they see a messenger RNA that they recognize because it has a motif, they go there and they inhibit the translation into protein. So the result is that there is less protein when the microRNA binds to this messenger. And the idea was that the Texel allele, the A allele, could be, that's the hypothesis, a microRNA target site for 3 different microRNAs, MIR1, MIR206, MIR122, while that would not happen in all the other wild-type animals which have a gene here. So to test this hypothesis, I would like to be very briefly here. I skip this. To be very brief, to test this hypothesis that there is a microRNA that is expressed in that cell that is interacting with the myostatin only in the Texel sheep because they have the mutation that generates a target site. We have to be sure first that the microRNAs are expressed in that tissue, in this case in muscular, in the myotubes of this sheep. We know that GDF is expressed, that we know. So we measure the expression of the microRNAs, and we see that all those among those 3 microRNAs, only 2, MIR1 and MIR206, are expressed in muscle. So those 2 microRNAs hold in the hypothesis, the other one drops. So the next step is now to see, okay, we know that the microRNA is there, that the messenger RNA is there, like the cat and the mouse. If there is more cat, there is less mouse. If there is microRNAs, and the hypothesis is true, there should be less myostatin. So we take Texel sheep and wild-type animals, and we measure, just focusing here, and we measure the protein levels of myostatin in serum, because we know that myostatin is secreted into serum, of Texel sheep and wild-type sheep. And I hope that is seen well. Yeah. I think you can see that the bands here, this is a Western blotting protein, that the bands here are clearly weaker than the bands here. Anyone can see that, isn't it? So that means that the serum of texel sheep has less protein than the serum of wild type sheep. So the hypothesis now holds very robustly. We have the texel sheep has microRNAs, these microRNAs have myostatin in the same tissue and has less myostatin. So the hypothesis is very robust now. Now we have to do one thing we have not done yet. Until now, what we did is try to be sure whether the hypothesis holds or not, but it was not a killing experiment, that will tell you yes or no. So that next experiment that we did was an experiment that would confirm whether this microRNA and this gene in texel and not in wild type interact. So what we did is a reporter luciferase assay. Very briefly, luciferase is a gene that when it translates into protein, it produces light, and you can measure this light with a camera and quantify the amount of protein that is there. All right. So what you can do is make a construct, and you can add a promoter, you can add a 3' UTR, any regulatory feature you want. So we make a construct with the 3' UTR of the texel, the A allele, and another construct with the 3' UTR of the wild type, and we put that into cells, and there we put the microRNAs, and we compare the expression. We can see, we can confirm that the interaction is happening. So you have a histogram here with a lot of bars. This is the amount of light that could be measured in the constructs. And I just would like you to center, if it's possible, in this two graph here. This for MIR1 and this for MIR6, 206. Those three here and those three here. This bar here is the construct with the wild type allele. You put the wild type allele, you put microRNA, and you look at luminescence. You see, this is this level. When you compare with the same construct but with the A allele, the texel allele, this is the light produced. Much less light, much less protein. Yes? So when we do the same for the other MIR206, the same, much less protein. Now, if we do it with two other microRNAs that we chose randomly, that we know that they, in the hypothesis, they do not bind this texel, this sheep myostatin, what you see is that the emission of light is not changing. Can everyone see that? No changes here, no changes here, a big change here, and a big change here for the microRNAs that are the hypothetical targets. So that study basically confirmed that microRNA 1 and microRNA 206 bind to specifically to the texel myostatin gene because they have an A allele, but not to the G allele, to the wild type. The next thing is, that's very nice, but only for sheep, and only for this example. The next question is, how important is that in life? Is that, because that's a very new laboratory element, nobody found that before. So is that happening only here, or is that really an important modulator of our phenotypes, all of us here, humans, other animals, sheep, plants even. So what we did is we studied the human genome and the mouse genome because there is, in those two species, there was already in those days, back in 2005, the human genome and the mouse genome was ready, was available, and quite a nice catalog of genetic variation was annotated there. So we searched all that and found that there is about 4,000 or 5,000 variations in each of those genomes, mouse and human, similar to the one we found, saying that this kind of genetic variation that affects phenotype through regulation of gene expression is very common. And we put that in a database that is called Patroclus, that is the one here. I think here is the website, www.patroclus.org, where all these things can be browsed. Nowadays, we have other species, we have cow, we have chicken, we have pigs, and you can also put your sequences and see your 3' UTR sequences and see if you have this kind of micro-binding sites. So it's quite a robust tool that is nice to use if your experiments require it. Now, briefly, we go to the second line of research I'm interested at, and this is what I did in London when I work in human genetics, which is search for the human psoriasis source 1 variant. It's very similar if you think, because the one was to look for a causal variant of texture muscularity. This is a taxal variant, a causal variant for a genomic locus that it has been known for many years that is very important in psoriasis. Psoriasis is a skin disease that affects between 2 to 3% of Caucasians, people from Europe, and its frequencies are different depending on the population. It's basically a chronic inflammatory skin disease, like patches like this. This is in the elbows. That affects keratinocytes, that involve keratinocytes, the cells on the skin, and T lymphocytes, immune cells. It's a complete disorder, meaning that there is many genetic factors and environmental factors. If you smoke, you have a much risk factor. It's a very important risk factor for developing psoriasis. And of course, it has a strong genetic component. So, before I showed you a QTL scan, microsatellites, F2 population, what I show you here is a GWAS scan, genome-wide association scan. This is in humans. This experiment was done like, let's say, 10 years later. And in humans, other resources, more availability of resources and technologies. So this is a GWAS that shows that this region here in chromosome 6 is a very strong risk factor. The higher it is, it's associated with the risk, okay? The higher it is, the more risk you have. So this bound here is in chromosome 6, the MHC measured histocompatibility locus that contains, is about 250 kb and contains 9 genes. Among them, the most candidate gene is HLA-C, it's a gene called HLA-C, and since the 60s, mid-60s, through serological markers, it has been seen that the HLA-CW6 form of that protein, mutant of that protein, is the strongest risk marker for psoriasis since the 60s. And this still now, with all the genetic polymorphisms and studies, still now the strongest marker is this protein itself. Okay? Important to say as well, it's very polymorphic, a lot of genetic variation in this region, and it's in high linkage equilibrium. That means that if you inherit the risk allele for the protein, all this chunk of the genome of 250 kb here, most of the times you will inherit all together because it doesn't recombine. So because this is very polymorphic, you will have a phase. If you have an A in a polymorphism here, and you inherit or a C, and the A is in linkage equilibrium with the risk allele in HLA-C, everyone that inherits the HLA-C risk allele will inherit as well the A. High linkage equilibrium. I hope you understand the concept. Okay. So the next thing is, okay, the most likely scenario is that source 1, as we call this genomic region, source 1 risk variant, the causal variant, what provokes this is HLA-C itself, the form of HLA-C. But because there is high LD with it, it is not stupid to think, it's highly probable that there is other polymorphisms that are functional, that have regulatory function, that are inherited together with the risk variant of HLA-CW6. What we want to find is what is the catalog of regulatory variants that could sum up the psoriasis effect in the source 1 locus. I hope that is getting clear. We're trying to find polymorphisms that are inherited together with HLA-CW6 protein and that are associated with the disease. Okay? To do that, because there is high LD, the first thing we need to do is to identify the variants that are in extreme linkage equilibrium with HLA-CW6, the protein itself, the risk allele for the protein. To do that, we have a list of sequences, and it's already something important that in the human genome we have a lot of sequences on nowadays, so that's a strong tool that we can use through bioinformatics. This is bioinformatics. Get data that is available in the databases, be able to mine this data to answer the question you have. Our question here is, there is a lot of genome sequence from human, and I want to know which variants are co-associated, co-segregate, are in high linkage disequilibrium with HLA-CW6. So I take all these variants, all these sequences, and I compare them, and I select those polymorphisms that have co-segregation with HLA-CW6, and I will show that in the next slide. Once we have done that, the next thing is take this list of variants and see which of them are likely to be functional. So again, you do bioinformatics. You take genome browsers and databases in which there is a lot of data from genome sequence prediction, people that has done predictions in genome sequences, from this to people that has done epigenetic studies or transcriptome studies, many things at the genomic level. We mine all that, try to see which of the variants that are in high LD with HLA-CW6 are as well in regulatory features, thereby being regulatory themselves. Okay? So very briefly, we generated from a psoriatic patient we did high throughput sequencing from the source one locus by some technologies that I won't explain now. And what we did is we took our sequence and we put them in that list. So we had our sequence and I think 15 more sequences. As you see here in the first column, there is 4 sequences from 4 different individuals from different studies, different research groups have sequenced that. So we have 4 of them that are HLA-CW0602. That means that we know that these people, in serological markers, taking blood and measuring protein, they had the HLA-CW6 protein variant. Okay? So that we consider risk sequences. And we take all the others from different haplotypes for HLA-C, 3, 5, 7, and we consider all of them non-risk. We compare all of them and we look for those polymorphisms that have one allele, for example an SNP, which is biallelic, an A or a T. We look for all these polymorphisms that have one allele, the same, in the 4 sequences that are risk, CW6, and absent in all the others here. And that means that they are candidates by presence in risk haplotypes. That's a word, haplotype. On those days, we get access to a high-throughput genotyping platform, something new, that was very dense, for specific regions related to immune-mediated disorders. And psoriasis is an immune-mediated disorder because it involves T lymphocytes. That platform really, in source one, had about 350 SNPs. This is huge. 350 SNPs in a region of 250 kb is more than 1 SNP per kb. Genotyped. That's a lot. We took 5,000 individuals from UK that were controls, not psoriatics, and we genotyped all them, okay? And look, linkage disequilibrium groups, again the same. If you inherit an A for this polymorphism, will you inherit, all the people that inherit an A here would always inherit the similar allele here, or they recombine? If they don't recombine, then they are in linkage disequilibrium, namely. So what we want to see is how many recombinations occur here. And this is what they call a heat map plot. The more red it is, means the more co-segregate, the more they co-segregate. I think those concepts are a bit difficult because it's genetics, but it's a bit difficult to explain that in 1 hour. Basically what it means is that these chunks of the genome are always inherited together. So what we see here is this black arrow is one allele that basically does not recombine, almost does not recombine. Everyone that inherits an allele here inherits always the same allele here. So this chunk has HLA-C and a pseudogene which is called HCG27. What we have done here is by this approach of linkage disequilibrium, narrow our region of 250 kb to a region of 179 kb. So now it's more narrow, okay? The next step that we do is look in this chunk, by the comparison I showed you before of sequences, which alleles are candidate, which alleles are always present in HLA-CW6 risk and absent in the others. And all of them are in this area here. It's about 144 variants, all located around HLA-CW6. So, by doing this approach, from an initial list of 5,000 polymorphisms annotated in the human genome, bioinformatics, you can go to the genome browsers and databases and you can retrieve this data, and you know that here there is about 5,000 common variants that are common in the population. Among them, we reduced that to 144. 44 reduction just by doing this. What we do next is, okay, now we have 144 candidate variants by presence in HLA-CW6 risk chromosomes and absent in non-risk chromosomes. So what do we do next? Which of them are likely to be functional? So we go again to bioinformatics and we mine databases to find epigenetic features that mark active or regulatory parts of the genome. So those are these three columns here. DHS means DNase 1 hypersensitive sites. STK4 monomethyl means a histone modification, so a methylation of lysine 4 of histone 3, which is associated to enhancers, and I will tell you what is that. And STK27 acetylation is an acetylation of lysine 27 in histone 3, marks associated with regions of the genome that are active. Those two guys here, they are chromatin marks, they are histones. And chromatin is the way that you find DNA into a nuclear cell, into a nucleotic cell. It's a combination of DNA with histones, with these proteins, and it's a way to map it. This is not just a packaging, it's as well, sorry, we know it has a function. So in this chromatin structure, when we find those modifications, that's indicating genomic activity, an enhancer, a promoter, these kind of things. An enhancer is like a promoter. A promoter is the region of a gene that regulates its expression, and it's contiguous. A promoter, and then the gene that is the part that is being transcribed. An enhancer is similar, but can be anywhere. And it's more difficult to predict because an enhancer, the prediction is you know where to go to look for it, near the gene. An enhancer you don't know, can be anywhere. Okay? So that's why we use this. We know that among our 144 variants, none of them maps in the coding sequence and change amino acid, none of them. None of them map into promoters, so we have to look for those other regulatory factors that are enhancers. DNase 1, DNA hypersensitive site, this is regions of the genome that are easy to break with restriction enzymes, and this by definition is also associated to regions of the genome that are open and active. So open means active, open means that transcription factors or the polymerase can get there and do the function. So we found 7 regions like this, and we selected 3 of them, what we call source 1 E1, source 1 E2, source 1 E3, because they had a redundancy on features. So that's very likely. Forget about this, I'm not going to get through that now. So we have 3 regions that are very likely to be real enhancers, to have regulatory function in our region of the genome, in source 1. But that was done by data mining, and data mining from other experiments that were done in healthy cells, in healthy patients, these cells from healthy individuals. So the question is that pattern changing in psoriatic patients? Because psoriasis have provoked some changes in the cells. So the pattern of enhancers that we found in healthy is very useful, but we need to be sure that it's the same in T cells, in psoriasis. So we did what is ChIP-seq, which is basically use antibodies to immunoprecipitate those histones that I showed you before. To immunoprecipitate these guys in chromatin from T cells from psoriatic patients. And we did all the monoprecipitation, and we did high throughput sequencing, we sequenced all that, and we mapped it against the genome. So again, bioinformatic tools to obtain sequences, and we identified peaks. Here, this is an intensity plot, which means the amount of immunoprecipitation of a genomic region. So you can do all that for the whole genome, and the higher it is, the more it immunoprecipitated. That means that the more histone, the more protein, in my case, histone modifications, was there. What I want to show you here is that this pattern is for T cells of psoriatic patients, and this pattern is for T cells of healthy controls. These patterns, and this is the same, this for HTK4 monomethyl, and this for HTK27 acetylation, the two histone modifications I showed you before, okay? What I can see here is that the patterns are almost the same. They are very, very similar. There is no different peaks here and here. The peaks are more or less the same. That's telling us that psoriatic T cells, the regulatory landscape in this region doesn't change, it's the same. Okay? So that means that what we did before is enough. We need to do it again to identify the regions that are regulatory, because we can see that this is the same between psoriatic and non-psoriatic. So the next thing was, I forget this one, I skipped this one as well. The next thing we did is to find, okay, so among our 144 variants, which ones map in those three regions that we found. We found DNase1 hypersensitive site, HTK4 monomethyl, HTK27 acetylation site here. So here, sorry, here, here, and here. Which of our 144 variants map in these regions? We found that only 9 of our 144 variants map in one of those regions, one in cancer 3. And among those 9 variants, which would be the guys here, among those 9 variants, 3 of them as well, again it goes, this is data mining, this is bioinformatics, 3 of them come up with transcription factor binding sites. This is regions of the genome that through experimental, through different experiments, people have seen that a transcription factor binds there. So a transcription factor is a protein that when it binds to the genome, it promotes transcription of a gene, okay? So what we found out is that among those 9 variants, 3 of them come up with small regions of the genome, very small, 20 base per long, most of them, that are transcription factor binding sites for transcription factors that are of interest in immune diseases, NF-κB, POV2, and other ones. So that stopped there. Now we are ongoing with other colleagues to do further analysis, but the idea here is that among the 5,000 variants that it was at the beginning in the database annotated, we have identified 3 that are very likely regulatory variants that are very likely as well to have an effect on psoriasis through regulatory features. And what we want to do now is to take one of these variants that affect our transcription factor, that is NF-κB, which is very important for immune diseases. We want now to put that into practice, into constructs, into cell constructs, and see if really interaction is real. So I know that was a lot of terminology for things that are unknown for many of you because you work in other disciplines. But basically what I say is that I have worked in human genetics, in animal genetics, and the idea is always to screen the genomes to find variants that have an effect in a phenotype, in a trait. Can be interest for livestock production, can be of interest for human health. And that these things have to adapt to new technologies, and nowadays technologies involve a lot of bioinformatics data because there's a lot of information out there that you can access online, and a lot of technologies to do high throughput sequencing and deep analysis. Okay? That's the only idea. Fine. So now then I moved to 1.5 years ago, I moved to, sorry, 1 second. I moved to the Center for Research in Agricultural Genomics in Barcelona. And this is a very interesting institute because it's almost unique in the world in the sense that it gathers genomics studies, high throughput genomics studies of plants and animals. Animals, mostly pigs, but as well sheep and goat a little bit, and we worked on rabbits some years ago. And plants, Arabidopsis as a model for genomics research, but also melon, strawberry, peach, and many, many others. I don't know all of them, a lot. And it has genomics resources for high throughput genotyping, real-time PCR for gene expression at as well as genomic scale, and for high throughput sequencing. What I want to do in what I'm doing already, and I want to do in the future, I have selected 2 experiments that I think can be more or less of interest as well for Egypt. And then I will briefly speak about 2 others in which Abdullah has been working with me. So the first one is this: try to understand a bit the molecular basis of canine leishmaniasis. Leishmaniasis is a parasitic disease that affects dogs, and it affects humans as well. So it's a zoonosis because it's a mosquito that that infects the parasite through biting the blood. Dogs and humans, but as well it can be horizontal transmission. Okay. The symptoms are a lot, and they are very ugly, and the animal and the person can die from it. It's not cured. The disease has no cure. Has treatment to alleviate the symptoms, but not to cure. Okay. And what I want to show here is that it's basically limited to the Mediterranean basin, more or less, and it affects the north of Egypt as well, the Mediterranean part of your country, right? So the thing is that colleagues of mine found by accident that toll-like receptor 2 is an immune gene, can be used, each protein can be used as a treatment, or is potentially interesting that could be used as a treatment in the future to treat that disease. They have seen that the number of parasites in the cells when you treat them with TLR2 drops. So what we want to do is to take a cell line from dog macrophages, because macrophages is the host cell. We want to take this cell line at 48 hours, I think. We transfect them, we infect them with the parasite, we wait several more hours, and this is being done by my colleagues, my immunologists, because this is not my area of knowledge. And at some point, 1 or 2 days later, they split and, in 2 different flasks, and in one flask they add TLR2, in the other they don't. We incubate several more hours or days, and then we harvest. Those that were treated with TLR2, those that were not treated, and also those that were even not infected. We harvest, and we extract RNA, and we extract chromatin, because that's for epigenetics. And we do RNA-seq, meaning that we sequence the whole transcriptome, and we do ChIP-seq, meaning that we sequence what we have immunoprecipitated, which is the same histones as before, one that marks enhancers, one that marks active regions on the genome, but here as well, this one that marks promoters, the promoters that everyone knows what they are, and another one that simply marks transcription. So it should be alongside RNA-seq, where it is being transcribed from genomic DNA to messenger RNA. Okay. We do all this, and then we compare them. We compare the 3 groups. But mostly this is the one we are most interested. We want to see what does TLR changes in gene expression and in gene regulation, enhancer, promoter, when you treat the cells. So I hope this next slide is not too complicated. This in the blue track, that represents genes. That represents, is a representation, that represents the whole genome, 25,000 genes. Imagine we found a subset of them that are differentially expressed between the cells treated with TLR2 and the cells that are not treated. Okay? That means as well already that there is a molecular difference. Something is happening when we treat with TLR2, that the gene expression, the expression of this gene and this gene changed. But not the others didn't change. Then what we do is we go to ChIP-seq pigs, basically we go to the patterns of enhancers and promoters of each of those cells, and we compare them. And each of them have a sequence. This is an enhancer. There is a sequence down there. We can take these sequences, and this is bioinformatics. We can take this sequence and read it to try to find if the system recognizes transcription factor binding sites again. And this is well known because there are systems that can do that. Can predict regions of the genome in which proteins that affect transcription, gene expression, bind. Those go, they recognize a location, they bind to it. So we can do that as well. We can use those tools. We take all those pigs, all of them, and we look at the sequence and we screen for transcription factor binding sites for all of them. And then we do something. For those that are near the genes that were differentially expressed, for those enhancers, for those promoters, near genes differentially expressed, we want to see if there is a transcription factor binding site predicted that is more frequent than we would expect. If we see 100 pigs like this, and 95 have a binding site for NF-kappa B, transcription factor, this is not happening by chance, because by chance we would expect 2 or 3, and we're finding 95 out of 100. Then we compare with the rest, with the whole of it, to see that this enrichment is unique to the differentially expressed gene regions. Okay? So what this is telling me is that probably NF-kappa B, because it's more present as a site, as a binding site than by chance, NF-kappa B, the protein, is the responsible of provoking these gene expression changes. So what do we next is to go back to the transcriptome and see if NF-kappa B itself was one of the differentially expressed genes. If yes, what we construct is an hypothesis. The hypothesis that we construct is that when you treat with TLR2 your cells, NF-kappa B levels change, and because NF-kappa B function is to regulate gene expression of other genes, the expression of those genes also change. And this develops something that promotes that the cells attack the leishmania. So what we are doing is identifying a molecular mechanism by which TLR2 can help attacking leishmania, and that can help us direct our treatments and our management of the disease, in this case in dogs, but as well it can inform human disease. So when someone with leishmania go to hospital, you can choose whether you give TLR2 or one of the genes that it prefers. So that would be one of the hypotheses, okay? Another example I wanted to put here is porcine cutaneous melanoma. With Abdullah, we have been as well discussing about it quite often. So, well, everybody knows what is a melanoma. It's a cancer of melanocytes in the skin. And there is many animal models. The one that is more widely used is the mouse, but the mouse is not a natural model. You have to provoke it through transgenesis, genetic modified organism, through xenotransplant, through irritation. You will not find a mouse that developed that spontaneously. So for us to study human melanoma, we need to challenge the mouse. The poor animal, we need to challenge him. So what we see, however, is that in pigs, there is some population of colored pigs that develop melanoma spontaneously, relatively frequently. But this is not being studied. There is one group in France, and another group that was in the US that is now in Spain, not mine, but somewhere else. But it's not really being deeply studied. So this is a resource that we're wasting just because for us pigs are food, but they are animals, and therefore their diseases are like ours, and we should study them. They can inform us. And we don't need to manipulate this pig, because that happens spontaneously. So the idea, well, the thing is that this is the most similar to human in the lesions, in the histopathology, in its progression. It has a lot of metastasis. It means that it spreads throughout the body, and this is what kills most of people in cancer, metastasis, as well the animals. But in these pigs, what is important is that it regresses spontaneously. It regresses alone. You don't do anything, and in a high proportion of the pigs, high proportion die. By high proportion of the pigs suddenly have a very strong immune response and attack the disease, and the animals cure, and they have a happy life until they have another future, another destiny. We know it's genetic, because we can trace family frequencies, and it has been studied a little bit in pigs, but not very much. And basically what I mean here is that the little bit it has been studied is already telling us that it resembles very much, as well in the molecular level, the human melanoma. And the idea here we want to do is to study this to see how good model of human disease, of human melanoma, it could be. And then with a special focus on the regression itself, because regression is very important, very interesting. If we could understand what is happening at the molecular level in these pigs, in the pigs that they cure spontaneously through immune function, and we could provoke that in human patients, that would be a fantastic therapy. So the idea that I want to do here is to do a GWAS, which is identify the genetic susceptibility to the disease. We take a lot of animals, some with melanoma, some without. We genotype a lot of markers in them. In this case, we genotype 60,000 SNPs throughout the genome, and we look for association. Which regions of the genome have risk? So if you have this allele, your likelihood to develop melanoma is 5 times bigger, 20 times bigger than if you don't have that allele. We want to find these regions through GWAS. So the only thing we do is go to the farm, and we take blood from 500 pigs. And the pig continues his life in the farm, so no economic cost for the farmer. We don't need to buy the pigs. It's cheap. It's a very cheap experiment. And then we do the GWAS. We identify the genetic region, the susceptibility. Then here we take, in the other color, we take 16 pigs, and this is for the functional study. So on that panel there, it's for the genetic susceptibility. This panel here is 16 pigs that we have in experimental farm, in our farm, to control them. And then here we do functional studies. We do again, like before, RNA-seq, the transcriptome, and chip-seq for the same histone modifications, epigenetics. And we collect all the data, we generate all this data to compare lesions. In this case, we don't compare animals. We compare for each of the 16 animals, within each animal, the lesion versus the healthy tissue. The lesion here is melanoma cells, malignant melanocytes. The healthy tissue here is healthy melanocytes. So we do RNA-seq and chip-seq for the lesion and for the healthy tissue for each of them. And we compare that. So we will find the differences, like I showed before in the leishmania, the differences in gene expression and epigenetic profiles between the lesion and the healthy. When we have all this data, the next thing we have to do is, okay, we know the genetic susceptibility, we know some molecular changes, but now important for us is to know, does this really resemble humans? Because if it doesn't, it's not a good model. We cannot compare carrots with potatoes. But if it resembles, then we can compare, because we know that the basis of both diseases, the human and the pig, are very similar. So we can use the pigs to study humans. And then we do that as well with regression, because at the same time it can inform us what is happening with regression, which in humans does not happen, and it can guide us for therapies. And in this regard, we have some contact with some people in the UK and in California that they work in immune therapy studies for melanoma research. So if we would succeed in the first bit and we would prove yes, they assemble, we would send them cells from our pigs for they to do their experimental tests with immune-based experiments. Okay, so there were two experiments I showed you that I think summarize very well what I want to do, and then I put two more in which Abdullah has worked when he was with me, and this is one is the genetic basis of NIFID, and NIFID is I cannot remember, it's neurofilament inclusion disease or something like this. It's a disease that affects the central nervous system in humans, and they have ataxia and they die very young. It's a very severe disorder, very rare, not frequent, very, very rare. One every 50,000, one every 100,000 people, very rare. But it's lethal. And there is no animal model, there is no mouse, there is nothing, so there is no way to study that well. The only way to study that is when we have necropsy samples. Okay? So some people in Barcelona were doing a mouse model, a genetic modified organism, for another study, for another thing. And when they do the genetics, they realize that the transgenics, they realize that the animal dies very soon because he has ataxias. So at the age of 4 months, they have ataxias and dies. So they cannot study what they wanted to study because it's a failed experiment. They wanted to do something and it's failed, it's doing something else. Something was wrong in the design. The guy goes angry, gets angry and he leaves. And he doesn't explain anything. The only thing we have is the mouse. We don't know anything else. We just know that it was done, he was targeting a region of the genome that harbors 2-3 genes. That's all the thing we know. So the hypothesis is, well, something happened wrong that affected the structure or the expression of one of those genes and developed the ataxia. So that what it means is that if this is true, so what they saw is that this mouse, actually this pathology resembles very much the NIFID in humans, the human pathology. So it could be a good model for humans, okay? So we study those genes in this mouse to see what was wrong during the transgenesis. And we do study as well human samples, genomic DNA from human samples, to see if that gene is also affected. Because if the mouse is a good model for that, and this is affecting one of these genes, until now we don't know anything about humans, but basically what we can know now is that maybe the same gene is then affected in humans, because this is what mouse is telling us. So we want to study the gene in human patients to see if they have a natural mutation, not transgenic of course, natural mutation that is provoking the disease. So that is the experiment. So we have been sequencing these genes in human and in mouse to find the mutations, and we have found now the transgenic error in mouse. We have to characterize it a bit further. We have not found anything in humans yet. But this is an ongoing experiment, and basically the idea is that we might find that this mouse is a good model for NIFID, and that at the same time is telling us what is the gene that is causing that in humans, because until now we didn't know anything. The second one is the genetic variation in swine taste receptor genes. Taste receptor genes, as everybody knows, we can perceive the taste of food in our tongue, okay? Sweet, umami, salty, bitter, sour. This is because we have some bugs that have taste receptors. But what has been known now is that taste receptors are present everywhere in the body, or in all mucosas. We have them in the brain, we have them in the lungs, in the airways, in the digestive system, and they have been associated strongly with toxicology. For example, it's known that if you are intoxicated by a toxin, you have increased expression of taste receptor genes in the stomach and decreased in the intestines. And what happens is that the stomach blocks its movements and the intestine enhances the movement. That means that if you have eaten a toxin, the body realizes, because T receptors tells the body, hi, you eat a toxin, I have recepted. In my receptor, I have noticed a toxin. The stomach will stop moving, so the toxin stays there, and the intestine will start moving so that you expulse it very fast. So it's a reaction mechanism. It's like the immune system that detects pathogens, but in this case it detects chemical molecules. What we want to study, so it has a very important function as well in diseases, all right? In diseases. So what we want to study is the genetic variation that is present in taste receptor genes in pigs. Again, because we work in animals. And when we have this catalog, all the mutations in taste receptor genes in pig, we want to genotype those that we consider more relevant in populations that have phenotypes on food and on health. Because that could help us then, if we find polymorphisms associated to food consumption, to health, it could help us first for the health bit as models for human diseases that are related to taste receptors, there are several, or for animal production, for pig production, select for different breeds, that's white, Europe, different breeds of pigs, select the diet that we know that will be the pig will like most. Because if the pig has a polymorphism that makes that he likes sweet better, we make a diet that is more sweet for this breed, but not for the breed that likes more bitter. Okay? So basically, we have selected all those genes. We have captured them, about 200 genes. We are sequencing that and did a big job selecting of those genes by bioinformatic analysis. Did a massive job there. And now we are sequencing that in a panel of animals to identify those variants. There are other things, but I think it's enough here. But at least I hope that you get an idea of what we do and what I'm interested in doing. Okay? Yes, that's it. Thank you.